So hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing Karnataka SET exam question paper for the subject Life Science. This is paper 2 questions that I am discussing. If you are new to my channel, please do like, share and subscribe. This test was conducted in 2024. And if you want still previous year question papers of Karnataka SET been discussed, you can go to my channel Omni Bioentrance. In the channel playlist, see for Karnataka SET or kset, and you will find them there. So coming to the next question, that is among the following processes, which is the major contributor of global warming? Generation and burning of solid waste, no. Contamination of water resources, no. Yes. Usage of fossil fuels, yes, that is the major contributor. Deforestation is also a contributor, but major contributor is fossil fuel. Then rice contain dash number of marker assisted selection genes, which is applied for the selection of pest resistant and nutrient rich strain, that is approximately 30. Then phytoremediation uses which of the following principles to immobilize and detoxify the pollutant? Okay. Now what is phytoremediation means? You will use plants to clean up the contaminated soil or water bodies or air. So these plants, they will absorb, degrade these pollutants. Okay. Example, if I tell you, it is in the case of sunflower, it will absorb lead, arsenic and cadmium from contaminated soil. Now there are various methods of phytoremediation. One is the phyto extraction. So these circles are your pollutants. Okay, phyto extraction. So circles are pollutants. So the plants will absorb these pollutants from the soil through their roots and distribute it to shoot leaves. That is extraction. Extract. Then there is phyto degradation or phyto transformation or even phyto conversion. They have mentioned here phyto conversion. So that means the plant, it will take up the contaminants from the soil and they will break them down into less harmful substances through various metabolic processes in the plant and also by using various enzymes present in the plant. That is phyto degradation. Then there is phytostabilization. So this is mainly done to lessen the dispersion of the contaminants in the soil. Okay, so what the plant actually do is it will sequester these pollutants within the soil matrix. How that can be by immobilize these pollutants by adsorbing and binding to them. See here the structure of the pollutant have changed after immobilization or the phytostabilization process. So as a result of this phytostabilization, the toxins will not easily disperse in the soil and re-enter in the other environmental bodies like water, etc. Okay, so it is done to prevent the soil from prevent the pollutant from dispersion and movement. That is phytostabilization. It involves immobilization of these pollutants by adsorbing and binding to them or by forming stable masses. Okay. Then there is phytostimulation. This is the phytostimulation. That is, it involves the breakdown of pollutants using plant enzymes as well as using the enzymes present in the microbes surrounding the rhizosphere of the plant. Then there is rhizofiltration. That is, it is the process of where the, or I can say where the roots will act as a filter and they will filter the water through these roots, and it will remove the toxic substances or the pollutants. The pollutants, either they can remain absorbed or either they can get absorbed by the roots, or they will remain adsorbed, adsorbed to the roots. That is rhizofiltration. And finally there is phytovolatilization. That means these pollutants which are taken up by the plant through the roots, they are transported to the leaves and the stem, and from the leaves they will be released into the air through transpiration. Okay. Now these are the various methods of phytoremediation. So phytoremediation uses one of the following principles to immobilize and detoxify the pollutants. So immobilize and detoxify means it can be through phytoextraction. Yes, there also you are taking in the plant is taking in the pollutants. After that it can go for phytovolatilization or phytodegradation. Again you are detoxifying the pollutant. Then phytoconversion, that is nothing but phytodegradation. Phytodegradation, yes, that is also correct. Phytostabilization, I told you here, where it is immobilized and preventing it from dispersion. So that is phytostabilization. So all 1, 2, 3 and 4 are correct. All these are methods of phytoremediation which will immobilize and or detoxify the pollutants. Okay. Then the blood that is measured to monitor stress and anxiety, the analyte. Okay. The analyte that is measured to monitor stress and anxiety using saliva is your cortisol. That is the mainly used, even though alpha amylase is also used, but cortisol is more correct, because cortisol it gives a reliable measure of the hypothalamic pituitary adrenal axis response to both physiological and psychological stresses. Okay, so cortisol, it is slower to get activated, cortisol production, because, and it is a reliable indicator because its level increases slowly and it will peak later, and it will remain in that elevated position state for a longer period of time. And alpha amylase, it is not like that. It is released in response to your sympathetic nervous system action, and it will provide a fast and immediate response to stress. Okay, it is a good marker for acute stress conditions, because in such cases when you apply the stress, the stress response rises quickly, and as soon as you remove it, it will come very fast to the baseline. Okay, it's a quick action mechanism, and it is useful for measuring the immediate physiological impact of an acute stress. So more accurate will be your cortisol one. Then there is another called as chromogranin A, and this is also a glycoprotein released by sympathetic nerve terminals and adrenal medulla in response to acute stresses. So the answer here is option 2. More correct is option 2. How does AFLP differ from other DNA fingerprinting technique like RFLP? AFLP uses DNA probes. No, RFLP is the one that is using DNA probes. AFLP use restriction enzyme. It is partly correct, but not only restriction enzyme. It uses a combination of restriction digestion, ligation to adapters, and a PCR amplification process. So 2 cannot be the answer. AFLP is limited to coding regions of the DNA. No, AFLP can be performed without any prior knowledge of the DNA of the organism's genomic DNA sequence. So that is also not correct. Then AFLP use selective PCR amplification and restriction enzyme digestion. Yes, that is correct. Whereas RFLP, it uses the DNA probes, then hybridize them to specific DNA fragments. That is the southern blotting technique is used there. Then, among the following option, which one is not true about fusion tags used in recombinant protein production? So, for fusion tag production, you in the expression vector, next to your target protein sequence, you will use a tag. And this tag, it will be a very small protein or I can say a short protein or a peptide sequence. And it will be added to your target protein sequence. So when this expression vector is allowed to produce protein, the tag will be attached to your target protein and you get what is called as a gene fusion product. Okay. Now, why do you use these gene fusion products in recombinant protein production is because it will help in solubility of the protein. So when you after cloning in the expression vector and you transform an E. coli cell, the recombinant protein, that is your target protein, it will not if it is not getting soluble in the bacterial cytoplasm, then it can accumulate in the form of inclusion bodies in the cytoplasm, and isolation and separation of these inclusion bodies will be very difficult. It will not give you the correct protein whatever you want. But once it is soluble means you can extract it from the bacteria very easily without much harm to your target protein. Okay. So that is what is meant by solubility. And maintaining in the soluble form means the functional state of the protein is also maintained. Now if it forms inclusion bodies, no chance. You won't get the functional state of the protein, or sometimes you may not get the required protein also. So that is why you require solubility, then stability of the recombinant protein, and in some cases if you are using chaperone tags instead of attached to the target protein sequence, then these chaperones, they will help in correctly folding of the target protein once it is expressed. Okay, now aids in identification of recombinant protein. Yes, if you are tagging it with GFP, then whenever you get a green fluorescence, you know your recombinant protein is expressed there in the organism. Okay, so that is correct. They have asked for the not true. Not true is being asked. Aid in proper folding. Yes, I told you if you tag with chaperones. Aids in improving the catalytic function. No, tags don't improve the catalytic function of the protein. Then aids in isolation of the expressed recombinant protein. Yes, now suppose you are tagging it with his. You are using his tag proteins. Then because of the presence of his tag, they will bind to the Ni2 plus column. So your recombinant proteins, they will bind to Ni2 plus column because of the presence of his tag in them. And after washing the column and by adding the eluting buffer, his tag will be what is that? His tag attached protein will elute out of the column after adding the elution buffer. You will get a pure sample of your recombinant protein. Then which of the following statement is true about type 2 error? So before coming to this type 1, type 2 error, I will tell you what is null hypothesis and what is alternate hypothesis. So null hypothesis, it is a statement that say that there is no effect, there is no difference, there is no relationship between the variables that you are going to work. Or okay, I am not telling work and confusing, okay, between the variables. Now alternate hypothesis means it is a statement that tell that yes, there is some difference, there is some effect, there is the relationship between the variables. Example I will tell you for null hypothesis is a new drug has been developed for blood pressure. So your null hypothesis means the drug will have no effect on the blood pressure. Remember, negative thinking and positive thinking. So negative thinking N, null hypothesis N. So no, 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 both. So that is null hypothesis. Okay, whatever you say the negative of that. No, it is not going to work out. That is null hypothesis. That way you remember, it will be easier. Then alternative hypothesis means it tell that yes, the drug is going to have some positive effect on the blood pressure. We will do the research in the lab and we will find out. So the researcher, he is positive minded. So he doesn't want the null hypothesis to be true. So his by default he will want to disprove the null hypothesis. Okay, now that is with regard to null and alternate hypothesis. Now come to type 1 and type 2 errors. So type 1 error is when you reject the null hypothesis when it is actually true. So by mistake, you have made a mistake and you are rejecting the null hypothesis when it is actually true. That means the example I will tell you, null hypothesis is you do not have the disease and alternate hypothesis is you have the disease. Now you do the medical test and there was some problem with the instrument for while conducting the medical test, and the test indicated that you are positive for the disease when you actually don't have the disease and you are healthy. So your actual situation is you are healthy, that is you don't have the disease. But what the researcher did, he rejected the null hypothesis, telling that you are having the disease based on the medical test he conducted. He didn't know that there was some error with the instrument. Okay, so that is type 1 error. Then type 2 error, that is here you failed to reject the null hypothesis. You failed to reject the null hypothesis when it is actually false. Confusion. Again come back to your hypothesis. Null hypothesis, you do not have the disease. Alternate hypothesis, you have the disease. A medical test for the disease indicate a negative result, that is you don't have the disease, but in reality you are sick, you have the disease. So here what you do is you fail to reject the null hypothesis. Okay, actually what the medical test gave result was you don't have the disease, so you went on accepting the null hypothesis. But actually null hypothesis is not correct and you are not rejecting the null hypothesis. Okay, or otherwise I can say you are accepting the null hypothesis, when in reality you are sick. Okay, you are accepting the null hypothesis. That is type 2 error. So type 1 is reject null hypothesis and type 2 is accept the null hypothesis. Now null hypothesis is also called incorrect hypothesis. So here type 2 error is accepting an incorrect hypothesis. Yes, that is correct. Then next one. In a positively skewed distribution, the relationship between mean, median, mode. Okay. Now skewness, it is a measure of the asymmetry of a probability distribution. So there is negatively skewed and positively skewed. So if it is zero skewness, then it is symmetric. So symmetric means everything perfect. Mode, mean, median, all are equal to each other. The data is evenly distributed around the center and there is perfect symmetry. See here, it is a perfect graph, symmetric distribution. Now come to positive skewness. Positive skewness, peak of the graph is towards the left and there is an tail extension to the right. So in this case, the mean is greater than the median, which is greater than the mode. Now example I will tell you is you have faced a very tough exam. What will happen? Majority of the students will score less. Few intelligent people, lucky people, more talented people will be there. They will get high scores. Very few people, not all, very few. Majority will be low score. Now what will happen? Most of the scores are low. So the peak will be towards the left. There will be a right tail extension. That is the few people who have scored some high marks. High scoring people. That is the tail. That is the right one. So it will, because of this few high scores, the mean will be shifted towards the right. So mean towards the right means, remember, mean is greater than median, greater than mode, and it is positively skewed. Remember this graph, mean, median, mode. From here you have to read. From right to left you have to read. Mean, median, mode. So mean greater than, median greater than, mode. Now coming to negatively skewed distribution. Here the peak is to the right, tail is to the left. Take the example of easy exam. What will happen? Majority of the students, they will have high score. Above and high score, above average and very high averages, they will score more. But a few of them, very below average, they will score less. So the shape will be majority of the scores creating a peak on the right side of the graph and the tail which is longer showing the low scores. So mean will be here on the left hand side. So mean will be less than, median less than the mode. It is negatively skewed. So the answer here is, it is for positively skewed distribution. So answer is option 2. Then when silver halide crystals are exposed to the radiation in an autoradiograph, which of the following chemical conversion take place? So silver halide crystal exposed to autoradiography, ionization is there. So this ionization, it will reduces the silver ions in the silver halide to metallic silver. So answer is option 3. Then image processing method after capturing image by microscope involves. So there are many image processing methods. I am discussing only image enhancement and restoration. So that is the answer. I will tell you what it is. So image processing after a microscopic capture means after capturing the image through a microscope, you try to enhance and analyze the image by adjusting the brightness, contrast, correcting uneven illumination, etc. All these processes are coming under image processing method of after capturing the image. Now one is image enhancement, that is adjusting the brightness and contrast so that the image is visible, perfect and evenly illuminated. Then image restoration means there is something called as degradation happening when you are capturing the image because of the blur and noise of the microscopic optics. Okay, so the first thing is mathematically you will remove this degradation, and it is called as modeling degradation. And once the degradation is modeled, you go for a restoration algorithm. All this is mathematics. Okay, restoration algorithm, so that it will inverse the degradation function of the captured image. What is the goal for this restoration is to produce a restored image that is very, very close to the original undegraded image. Okay, that is the means of this restoration technique. Then next one, EEG is conducted to analyze the following except perform brain activity recording, yes. Diagnose conditions affecting the brain, yes. Direct epilepsy, yes. Brain tumor, no. It is not conducted to analyze brain tumor, although in the early stages of screening they use EEG, but it is not the principal tool for analyzing brain tumor. In early screening they do because certain physiological changes a tumor can cause in the brain you can detect by EEG, but anatomical changes will be visible only in an MRI. So for precise location and diagnosis of brain tumor, you usually prefer the CT and the MRI scan. They are the principal tools for brain tumor detection. Then last question, which of the following method is not used for ground sensing? So ground sensing, it is a method of data collection and using sensors by placing these sensors at or near the earth surface, and you get details for mine exploration, hydrogeology, monitoring soil contamination, groundwater monitoring, landfill monitoring, archaeological monitoring, etc, etc, etc. All these things you can monitor by using ground sensing methods. So there are various methods for ground sensing, one is not used. So we will see ground penetrating radar or the georadar. So it uses radar pulses to image the subsurface structures of the earth. Then electrical resistivity tomography, it is used for mapping geological features, groundwater pathways, etc. Then there is magnetometry, which measures variation in the earth magnetic field, and it detects buried metal objects, archaeological remains, magnetic minerals, etc. Okay. Then there is seismic survey, which is used for oil and gas exploration, etc. So what is not used? Visible and infrared sensors. So that is your answer. So thank you for watching my channel. This is the end of the question paper. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of Karnataka SET being discussed, you can go to my channel Omni Bio Entrance. In the channel playlist, see for Karnataka SET or kset, and you will find them there. Thank you.